Hi guys, welcome to my channel Pharma and Fun. Here is my first video on the pharmacology introduction lecture. This lecture is very important for your vivas and also to develop basic concept about pharmacology. I hope you like my videos. Kindly like my videos, subscribe my channel, share my videos, and never forget to click on bell button. And the most important thing, I'll upload my videos on Saturday, Tuesday and Thursday. Here I'm going to explain you definition of pharmacology. The pharmacology from the Greek pharmakon means drug and the logos means treatise or discourse. Pharmacology is the body of knowledge concerned with the action of chemicals on biological systems. So the important thing is how the chemicals act on biological systems. Broadly we can define as how chemical agents affect the living processes like hormones, neurotransmitters, growth factors, drugs, toxic agents in environment. So basically pharmacology deals with the chemicals which acts on biological system, which acts on the living process. We all know about the pharmacology is revolving around drugs, and these drugs are the chemicals. So here it is a complete definition of pharmacology. Whenever someone asks, you can explain it. In the next slide, I'm going to discuss you about what is drug. I remember when I was in first prof, one of my teachers asked the definition of the drug, and the most of us were like how we can explain the drug. So here I mention a brief definition of drug. The drug is a chemical substance present in medicine that is used for diagnosis, prevention and treatment of disease. So here I emphasize on the chemical substance. The drug is a chemical substance, or the purpose of drug is diagnosis, prevention and treatment of disease. The WHO explains in 1966 like the drug is any substance or the product which is used or intended to be used to modify or explore physiological system or pathological states for the benefit of the recipients. So here we can see that the two terms we use, the physiological system or pathological state, how the drug modify or explore these systems and affect on pathological state. Pathological states mostly deal with the disease condition. So we know that we take the drugs or the chemicals when there is uncertain conditions in the body. So the drug which affects on pathological states or modify the physiological system, and all these things we are doing for the benefit of recipients, and the recipients may be animals or maybe humans. Here I mention a flowchart which explains the nature of drug. Whenever someone asks the definition of pharmacology, you should mention the two terms like pharmacodynamics and pharmacokinetics. These are the two branches of pharmacology. The whole pharmacology is revolving around the pharmacodynamics and pharmacokinetics. Here the pharmacodynamics explains like two more things, like two things under the pharmacodynamics, which is receptor site and inert binding site. Pharmacodynamics which is inside the receptor site and inert binding site. I will explain the definition of pharmacodynamics and pharmacokinetics later in later slides, but here I mentioned the flowchart which explains that pharmacology has two branches like pharmacodynamics and pharmacokinetics. Pharmacokinetics mostly deals with movement of drug in body. The most important term is ADME. In next slide I mention what is ADME. A is equal to, A means absorption, D means distribution, M means metabolism and E means elimination. So pharmacokinetics deal with how the drugs affect in body, how it causes the absorption, distribution, metabolism and its elimination. So this flowchart explains drug development and regulation. The drug development and regulation, how the drug develop and then regulate. First of all, we check the safety and efficacy of chemical agent. These chemical agent is, we can say the drug is toxic when we give above dose. The drug is a chemical substance. A chemical substance can be a toxic when we give above the dose, when we give a high amount of dose. So dose is the major thing that convert the chemical agent a drug, otherwise the drug is a toxic. So for this we have to check the safety and efficacy. Then we done with animal testing, clinical trials, patent and generic drugs. Then this drug come into the market. Here I mentioned some important definitions. These definitions are very important for your vivas. Like drugs, I already mentioned that explain the drugs definition. Drug receptor. When you take some medicine or a drug by mouth or some, it goes inside your body. What happens? The drug binds to the receptors. Then it produces an action. Otherwise, the drug can't produce an action. For producing an action, the drug have to bind some receptor. And what is the receptor? Receptor is a molecular component of the body with which drug interact to bring about their effects. Like, we should know what is the receptor is. Receptor is a molecular component of the body with which drugs interact to bring about their effects. Okay, molecular component of the body with which the drug interacts to bring about their effects. So to produce the effect, the drug has to bind with the receptor. What is the distribution phase? Distribution phase, the phase of the drug movement from the site of administration into the tissues. Like distribution phase in which the drug distributes, where the its movement from administration into the tissues where it distributes. Elimination phase is the phase of the drug inactivation or removal from the body by metabolization. Once the drug does all this action, the drug is converted into inactive form and then it removes out from the body. Here is the endocytosis, exocytosis, endocytosis. These are the terms which is when the drugs goes inside the cell, which is endocytosis, and then the expulsion of the material from the cells or the vesicles, which is exocytosis. So you can take the definition of from these slides. The important things which is the definition of pharmacodynamics and pharmacokinetics, which is what is the difference between the pharmacodynamics and pharmacokinetics. Pharmacodynamics, the action of a drug on the body. Okay, what is the pharmacodynamics? Pharmacodynamics is the action of a drug on the body, it including receptor interaction, dose response phenomena, and mechanism of therapeutic and toxic actions. So important thing is dynamics deals with action of a drug on body. What does the drug do on the body? What does the drug produce an action on the body? These things include receptor interaction. I already mentioned the drug produces its effect when it binds to the receptor. So receptor interaction, dose response phenomena, or mechanism of therapeutic and toxic actions. Pharmacokinetics deals action of the body on the drug. What does the body do with the drug? Dynamics, what the drug do with the body, what the drugs do with the body, and the kinetics, what the body does with the drug. So it includes ADME. ADME, A for absorption, D for distribution, M for metabolism, E for excretion. At this point, you just know the definition and the basic concept. And when you complete the whole pharmacology, you automatically understand the absorption, distribution, metabolism, excretion phenomena. Biodisposition is a term sometimes used to describe the process of metabolism and excretion. Okay. In pharmacodynamics, these points include in pharmacodynamics. Whenever someone asks explain the pharmacodynamics, you can explain the definition and these points like receptor effectors, dose response curve, agonist, partial agonist, inverse agonist, antagonist, signaling mechanism, receptor regulation. These definitions about agonist, partial agonist, inverse agonist, antagonist. I will upload a next slide in which explain these definitions. So these definitions are very important, also very important for your bio point of view, like what is the agonist, what is the partial agonist, what is the inverse agonist, what is the antagonist. So I will explain these definitions in the next slide. I hope you will like those too. So in the next slide, here the very important question about and the basic of pharmacology, like what are the receptors, definition of receptor, and what are the types of receptors. This is the big question of pharmacology. The first question, I remember this question always my examiner asks this question in each and every paper, like what are the types of the receptors. So here I mentioned the types of receptors like G. There is a total 1, 2, 3, 4, 5, 6 receptors, and the most important one is GPCR receptor. The second one is ion channel, and third one is transmembrane enzyme. Fourth is transmembrane non-enzyme, then nuclear receptor or intracellular enzyme. So there is a functional family of these receptors: physiological ligands, effectors and transducers, example drugs. So the source of my slides are Goodman, Gilman and Katzen. If you wanna complete explanation of these receptors, you can tell me in your comments. I will explain the mechanism of each receptors in detail, but here I mentioned some basic mechanism of the important receptors. Okay, so but you can study on the Goodman-Gilman, which is the very I have taken these receptors or the content from the Goodman-Gilman. They explain everything in a very easy way. So functional families, you can say beta adrenergic receptor, muscarinic cholinergic receptor, eicosanoid receptors. You don't take any tension about these receptors. You should know at this point how many types of receptors and what are the receptors. Okay, when you complete your second or third chapters of pharmacology, then you automatically understand that what is the muscarinic families, what are the cholinergic receptors, what are the and these receptors belong to the GPCR. Examples of the drugs: dopamine, propanol, atropine. At this point you just take this and have a look, and the next slides you will understand and learn the definitions. Okay, so in the next slide I'm going to discuss about the G protein coupled receptors. This is the most important receptor. First of all, I will explain it from the diagram. In this diagram you can see there is a basal body, alpha, beta, G protein coupled receptor. There is 3 subunits of G protein coupled receptors, that is alpha, beta and gamma. When the alpha subunit is binded with the GTP, okay, when there is a ligand attached to the receptor, this GTP release from the alpha subunit and convert into the GTP when the receptor is active, when it is bind to the ligand, and this active GTP is bind to the alpha subunit. When it bind to the alpha subunit, it releases the beta and gamma subunit, which is the modulation of factors. It's called the modulation of factors. At this process, one of the protein RGS released and PO4 releases. Ligand binding stimulates, GTP releases, GTP binds to the alpha. Active alpha GTP returns to basal state by increased RGS protein. So we can discuss it from here. Diagram showing the stimulation of a G protein coupled receptor by ligand, the activation of G protein and stimulation of selective factors. Systematic diagram of the mechanism involved in the control of cell function by G protein coupled receptors. G proteins and effectors. In absence of ligand, the receptor and G protein heterotrimers form a complex in the membrane, with the G alpha subunit bound to the GTP. Following binding of ligand, the receptor and G protein alpha subunit undergo a conformational change leading to release of GTP, binding of GTP and dissociation of complex. The activation of GTP bound G alpha subunit and the free B gamma dimer bind to and regulate effectors. The system is returned to basal state by hydrolysis of the GTP on the alpha subunit, a reaction that is markedly enhanced by the RGS protein. Prolonged stimulation of the receptor can lead to down-regulation of the receptor. Down-regulation means the number of receptor decreases, so there is effect decreases. This event is initiated by G protein receptor kinase that phosphorylates the C-terminal tail of the receptor, leading to recruitment of protein termed elastin. Elastin binds to the receptor on the internal surface, displacing G protein and inhibiting the signaling. So in this, whenever someone asks the GTP receptor, so you can draw this diagram which clearly explain how it affects. When the ligand bind to the GTP, it becomes active and alpha GTP is converted to delta GTP. B and gamma subunits releases. When alpha to normalize its effect, the alpha GTP, the RGS protein, when RGS protein increase, this alpha GTP is hydrolyzed and the GTP is again converted into the GTP. So this is a cycle. Modulation of factors is done by the beta gamma and by phosphorylation and the scaffold proteins, which is elastin. So you can see how the gamma beta again bind to the alphas effectors, and which increases the effectors and modulators like adenyl cyclase, PLC, and this is a modulation of factors, how the effectors modulate when it's down regulated. The second important receptor is ion channel receptors, and there is a types of ion channel receptors. The most important types of ion receptors are two, which is ligand gated channels and voltage gated channels. But here I mentioned four. Leakage non-gated channels are always open. Nerve cells have more potassium than sodium leakage channels. As a result, membrane permeability to potassium is higher. Explain resting membrane potential of minus 70 millivolt in nerve tissues. Ligand gated channels open and close in response to the stimulus. Whenever there is a stimulus, these channels open and then close, open and then close. Results in neuron excitability. Voltage gated channels respond to a direct change in membrane potential. Ligand gated channel, when the ligand binds, these channels open. When the ligand is not, these channels closed. Ligand act as a stimulus. The voltage gated channels act as a membrane potential. Mechanically gated ion channels respond to the mechanical vibration or pressure. So this is a picture of voltage gated channel. When a stimulus comes, it's open and sodium ions go inside, and this is the activated channel. And when there is no stimulus and the sodium ions cannot go inside, this is the inactivated state. And the drugs mostly undergo this mechanism is carbamazepine, phenytoin, lamotrigine, and valproate. Ion channel receptors are ligand gated ion channel receptor acts as a gate when the receptor changes shape. When a signal molecule binds as a ligand to the receptor, the gate allows specific ions such as sodium or calcium through a channel in the receptor. Ion channel receptors are very important in the nervous system. So I hope you understand what is the ion channels. Whenever there is a ligand bind, it opens and then sodium and calcium ions go inside and effects occur. These are the things like this, how the signal transfer, how the nervous system act. So mostly the nervous system undergo the ion channel receptors. This is the basic mechanism. When in the later chapters of pharmacology, when we study in detail about the autonomic nervous system and when we study the detail about the autonomic nervous system, so you can understand how the drugs acts on these ion channel receptors. When the channels are closed, the ions cannot pass through membrane. When the channels are open, the ions pass through the membrane. And we can use this natural thing, we can modify it or inhibit it by using the medicine. There is a medicine which enhance this action and some medicine which inhibit this action. So at this point you just need to understand the receptors and their mechanisms. The next receptor is enzyme-linked receptors. The enzyme-linked receptors function both as enzymes and as receptor. And there is some enzymes in the membrane, so these receptors acts on the basis of that enzymes. These enzymes are basically transmembrane protein, with the receptor side facing the interstitial fluid and the enzyme side facing the cytosol. Most enzyme-linked receptors are tyrosine kinase, others are guanylate cyclase, which cyclize the conversion of GTP to the second messenger CGP. These enzymes are normally inactive but are activated when messengers bind to the receptor, which allows them to catalyze intracellular reactions. Okay, this is the important thing. This is the mechanism. Like mostly these enzymes are inactive, but when there is a messenger when there is some signal the messenger bind to the receptor, they become activated and the enzyme produce their action. A messenger binds to the receptor, changing its conformation. The conformation change activates the tyrosine kinase. The tyrosine kinase then catalyzes phosphorylation of an intracellular protein. Phosphorylation of the protein changes its activity by covalent modulation, bringing about a response in target cell. So this is the mechanism of enzyme-linked receptor, which is very concise and brief. Mostly these receptors are inactive. When the messenger binds to the receptor, it becomes active and changing its conformation, and conformation change activate tyrosine kinase receptors, and these activation convert the protein, catalyze the phosphorylation and release the ATP energy response to the cell, alter metabolism, regulate proteins synthesis by covalent modulation, bringing about a response in the target cells and produce a response in the target cells. I mentioned the most important types of the receptors like GPCR, ion channel, enzyme-linked receptor. If you want to get more information about these receptors, you can comment on this you can comment at this video at the below, so I can explain it more. So this is my first video on the pharmacology lecture, which is the very basic and very important, that deals with the concept of the pharmacology. I hope you like my videos, so never forget to like my video, subscribe my channel, share my video, click on bell button, which is very important. You can comment me your topics. I will upload the next video on Saturday, Tuesday and Thursday, which is most likely maybe on the autonomic nervous system. That is very important to understand. So I hope you like my videos. Thank you for watching. Bye bye.